For the video, n equals 3, which means there are 3 pairs of chromosomes. The red chromosomes are maternal and the yellow chromosomes are paternal. We have different lengths of chromosomes to achieve n equals 3. We are going to talk about mitosis and meiosis. Metaphase is the third phase of mitosis. In our picture, the sister chromatids align at the middle of the cell. Sister chromatids are 100% identical to each other. There are spindle fibers that divide the sister chromatids into daughter cells. Metaphase is important because the sister chromatids are ready to divide into daughter cells.
Anaphase is the fourth phase of mitosis. In our picture, the sister chromatids are divided into daughter cells. Daughter cells contain the same number of chromosomes as their parent cell. The spindle fibers divide the sister chromatids into daughter cells. Anaphase is important because the sister chromatids are pulled apart to either side of the cell, which will lead to 2 diploid cells. Diploid cells are 2 copies of each chromosome. Crossing over is when the identical length segment of DNA switches between 2 homologous chromosomes, which creates genetically distinct sister chromatids. Prophase 1 of meiosis performs crossing over when the homologous chromosomes pair and exchange segments.
The first round of meiosis splits the homologous chromosomes. Homologous chromosomes are identical sequences of genes. In metaphase 1 of meiosis, the pairs of homologous chromosomes align in the middle of the cell. The tetrads line up, which means that the homologous pairs bind each other and there are spindle fibers. Metaphase 1 is important because the daughter cells only have half the number of chromosomes of the original cell. Each daughter cell is a haploid, which means that there is only 1 copy of each chromosome. After metaphase 1, the next cycle is anaphase 1. Anaphase 1 splits the pairs of homologous chromosomes. 1 chromosome of each pair moves to one side of the cell, and the other chromosomes move to the opposite side. Anaphase 1 is important because it makes sure the sister chromatids are separated into 2 equal sets.
Independent assortment happens during meiosis. The homologous chromosomes assemble in tetrads, and each homologous pair splits randomly and independently, which means that there are 2 to the third combinations of homologous chromosomes. Anaphase 1 of meiosis performs independent assortment. Meiosis 2 separates the sister chromatids. In metaphase 2 of meiosis, the spindle fibers align with the sister chromatids of each chromosome. The spindle fibers are attached to the centromere of the sister chromatids. The centromere is the point where the sister chromatids are connected. Metaphase 2 is important because it prepares the sister chromatids to separate in the next phase of meiosis.
After metaphase 2, the next phase is anaphase 2. In anaphase 2 of meiosis, the sister chromatids separate and move to the opposite sides of the cell. The sister chromatids migrate along the spindle fibers into the opposite poles of the cell. Anaphase 2 is important because the sister chromatids finally separate, which will lead to 4 haploid daughter cells. Most prokaryotic cells divide by the process of binary fission. In eukaryotic cells, cell division mainly occurs in mitosis and cytokinesis. Binary fission is a form of cell division in prokaryotic organisms that produce identical offspring. Cell division in eukaryotic cells includes the nucleus dividing, which is mitosis. Next, the cytoplasm divides, and the daughter cells form, which is cytokinesis. Cytokinesis is the cell membrane pinched to create separate cells. To clarify, cytokinesis is not part of meiosis, but happens at the same time.
Mitosis involves 1 cell division, and meiosis involves 2 cell divisions. Mitosis creates 2 genetically identical diploid cells, and meiosis creates 4 genetically diverse haploid cells. The purpose of mitosis is the growth of cells, repair of cells, and asexual reproduction. Asexual reproduction involves one parent and produces offspring that are genetically identical to each other and the parent, a clone. The purpose of meiosis is genetic diversity and sexual reproduction. Sexual reproduction would have more diversity, which leads to more opportunities for survival and reproduction. Thank you for listening to our video.